So, good morning all, good afternoon from those who are outside Brazil. Thank you for joining us, for coming for my PhD defense. So, I'm Isabella, I'm a double degree student here at the University of São Paulo, Brazil, and at the University of Copenhagen, Denmark, from the double degree agreement. And today I will share with you some of my results of my thesis entitled Virulence of Metahizio anisopli and Metahizio Riley propagulis, a molecular investigation during the infection on different insect hosts. So I will start with some background just for you who doesn't maybe don't work with biological control, just to put all of you at the same page of what we are talking about. This is the background of the biological control in Brazil. And we have all the products here. Many of them are microbiologicals. Most of them, actually more than 60% are microbiologicals products. We have macrobiologicals, insects and predators, parasitoids, biochemical products, and semiochemical also. Of those microbiologicals, most of them are bioinsecticides. We have biofungicides, bionematicides, pheromones, and growth regulators. Of those bioinsecticides, we have a problem here, which is the many biological products, special bioinsecticides in microbiological, they are not registered, and of those who are registered, here we have only the registered, they have low technological level, many of them are the same isolates or the same species producing all over the time, as we can see here. We can see an improvement of these registered products on the last years, but they are still need improvement of knowledge and technology. So among those insecticides, we have the entomopathogenic fungi. They are the commonest pathogens of insects and regulators of pest populations. They can be produced in vitro. That's why they are improving production and selling of this kind of products for use as biological control agents in large scale on the field. And on the last few years, we had an improvement of production and formulation technologies, which could put more products on the market, and they are attractive components for integrated pest management practices. Here, just for example, the infection process and which propagules we have on entomopathogenic fungi. So we have first the conidium. This conidium adheres to the insect cuticle. So this picture here shows the insect cuticle. The conidium, the spore of the fungi, adheres on this cuticle. It forms the germ tube, forms the appressorium, who makes pressure, physical and chemical pressure here to penetrate, form blastospores inside the insects. Those blastospores will colonize the insect and form mycelia and go outside the insect. So this is a quick and easy way to understand how those entomopathogenic fungi act on the insect. So we have blastospores here on the right. Those blastospores can be produced in vitro, but they are formed inside the insect, inside the hemolymph. And here on the left, we have conidia, aeroconidia, which is produced outside the insect on the surface of the insect. Most of what we know about those interactions between fungi and their hosts, the insects here in our case, has been determined with the Hippocrealium and Ascomycetes, Metahizio and Beauveria. Most of the studies are done with those two genders, Metahizio and Beauveria. These species are commonly applied as biopesticides. They can colonize plants, can be used as plant endophytes for improved agricultural production and also for biological control of pests on the field. The metarhizium is the aim of our study. It is high abundant and globally distributed. It is best known for infecting and killing arthropods, but it can switch between different lifestyles. That's why we are talking about different species and different lifestyles and different ways to colonize different hosts. They can be found as saprophytes on the rhizospheres and as endophytes as well. At the end, they can be broad or narrow growth range. They can be generalists or specialists among those species. So talking about speciation and host adaptation, so we have the generalists and the specialists. The generalists, they have mechanisms to invade the host. They are less adapted to deceive the immune response to kill the host, so they need to kill quickly. Usually those species, the generalists, they produce more enzymes to kill really the insect fast because they cannot trick the immune response of the host. The specialists, they are, as the name tells, specialized on the host, on the insect, so they know how the insect works. They are adapted to invade and trick the host immune response, and they don't need to kill quickly. Usually they have different mechanisms to evade this host, and they don't need to have many enzymes to kill fast. So we have comparing here Metarhizium anisopli and Metarhizium riley. Why? We choose the Metarhizium genera, but we have anisopli and riley, where one is generalist and the other one is a Lepidoptera specialist. So the Metarhizium anisopli, it is produced, the blastospores of Metarhizium anisopli is only produced in liquid media, and the Metarhizium riley, it produces its cells in solid and in liquid media. The host range for Metarhizium anisopli, they are generalists, as I said. Metarhizium anisopli is a specialist. And another difference between anisopli and riley, Metarhizium riley can be epizootic in field populations for Lepidoptera species, and Metarhizium anisopli, no, he cannot be epizootic. And regarding commercial production, is important things to say here. We can produce Metarhizium anisopli in large scale, but we are not able yet to produce Metarhizium riley in large scale. So for this thesis in general, we have this first hypothesis, which is the liquid media with high osmolarity produce high yields of smaller and more effective blastospore of Bovera batsiana, which is a paper from Mascarin. And we would like to test if osmotic pressure can really improve the mass production of Metarhizium in liquid culture fermentation, producing higher yields of smaller and more virulent blastospore. So if it can do it with Bovera bassiana, why not with Metarhizium species, as they are at the same Hypercorales group. And besides that, we have two species, Metarhizium anisopli and Metarhizium riley, that evolved differently. So they are really different, but they are among the same genus. And we have two different reproductive structures. We have blastospores and we also have conidia. And the transcriptome studies could lead us to understand, first, if Metarhizium anisopli and Metarhizium riley use different mechanisms to penetrate Spodoptera frigipeda, a caterpillar. If blastospores of Metarhizium anisopli and Metarhizium riley are more virulent than conidia, for example, and if Metarhizium anisopli can also modulate its phenotype and gene response to adapt and cause disease to the different insect orders. Just because Metarhizium riley is a specialist and Metarhizium anisopli is generalist, this generalist is able to penetrate and infect different hosts. We know, yes, but how did he do it? So the thesis summary, in general, the first chapter is a general introduction where I told you our hypothesis, a brief history of what we are talking about, our hypothesis, our main questions. Then on the second chapter is a review where I talked about the genomic perspectives, look into the evolution on fungal host specificity, discussing generalists and specialists in metahism genera. Then on the third chapter, I talked about the blastospores from Metahism isopli and Metahism riley, and we look into how they penetrate and how which genes they express in each case. And on the last one, we investigate the phenotypical plasticity, looking into transcriptome profile of Metahism 1 isopli during penetration in different kinds of insect wings. So I will start with the chapter 3. I will look into chapters 3 and 4, where we have many results to discuss. This paper is already published in Microorganisms. And starting with Metahism 1 isopli, I already talked about, but just to put you on the context of this study. So the Metahism 1 isopli is well applied, especially here in Brazil. It is globally distributed, but here in Brazil is well applied, and we have one of the best cases of biological control for spiral bugs in sugarcane. And we also have Metahism riley that is not commonly applied on the field, but it causes natural epizootics in Lepidoptera species, especially in soybean culture here in Brazil. And we have two fungal propagules. We have Conidia and we have blastospores. The difference between them, apart from where they are inside the insects, the Conidia are hydrophobic. They form uniform cells. All of them have the same size. They can be produced between 12 and 20 days in solid media, well produced in solid media, and it has higher shelf life. The blastospores are hydrophilic cells. They are pleomorphic. They have different sizes among them. The production is really fast in liquid media, so we cannot produce blastospores in solid media, only in liquid media. And it also has higher virulence. But little is known about the factor that allowed these blastospores to infect and kill insects, because originally they are formed inside the insect. So why they can be produced so fast, they can germinate so fast outside the insect, you know? And we also have the Spodoptera frigipeda, which is an important pest crop here in Brazil, all over the world, but especially here in Brazil, and can attack corn, sugarcane, sorghum, and soybean. And the hypothesis and objectives of this paper, we have the hypothesis of the glucose concentration, which improves mass production for our hypothesis. It improves mass production of Metahism species in liquid culture fermentation, producing higher yields and smaller and more violent blastospores. And we compared the blastospore yield, the size and the virulence to Spodoptera frigipeda after being produced using those different conditions of osmolarity. So a normal condition of osmolarity and a high condition of osmolarity. We use Spodoptera frigipeda because the Metahism riley is a specialist in Lepidoptera, so we need to choose one specialist to make the tests. And the second hypothesis is the transcriptome profile of blastospores must change during the cuticle penetration, being different among Conidia and blastospores. So if they are so different, they must be adapted differently to insect hosts. So for that, we characterize the gene expression profiles of those propagules, Conidia and blastospores, on the caterpillar cuticle. So this is a little bit different of others papers which studies production on insect segment, because other papers studied production in insect tegument, but making a substrate of the caterpillars. Here we study inside the tegument of the real caterpillar. So first we reproduce those blastospores in liquid fermentation. We use the Jackson liquid culture medium. We use two high of carbon source. We use the flasks with the medium, and we put on the shaker for 3 days. We use 2 species, the Metahizum anisopi and the Metahizum riley, and we choose 2 different isolates from each one for those first tests. All the flasks were checked, and we plated them to avoid any contamination and to be sure the isolates was correct and we had no contamination for those studies. And we measure the blastospores volume. So we use the Leica system besides the microscope to measure the length and the width of 50 random blastospores of each treatment. We used 4 replicates for each one. And then we performed the virulence test of those blastospores produced on different higher of osmolarity. We used Potter Tower, and we put those caterpillars individualized into those trays to analyze it. And after mortality, we put on the humidity chambers to confirm the mortality with the sporulated. The survival analysis was using the parametric model for survival data, the Weibull distribution. Then, before the RNA extraction for transcriptome studies, we did the microscopic, the scanning electron microscope to know the exactly time after spraying the blastospores or the conidia are germinating and penetrating, because we would like to know the exact time for those RNA extraction to be sure we will take a picture of that exactly moment. So we performed those images using blastospores 2 hours, 4 hours, 6 and 8 hours after spraying. And for conidia, we already know conidia usually germinate between 18 and 24 hours. So we did it 12 hours, 16, 24, and 32 hours, just to be sure that exactly time the apressoria was being formated and the penetration occurred. So for those studies, we selected only 1 isolate for each species, the ESALC E9 and the ESALC 5611. So we performed the RNA extraction after the results of the microscopic studies. And the RNA extraction was performed using the cuticle. So I sprayed here the fungi, and then I took off the digestive tube and used just the cuticle of the caterpillars. And the treatments was blastospores, conidia from Metahizum anisopi, and blastospores and conidia from Metahizum riley. So here is what I did in general. So I sprayed here inside the caterpillar. I took only the cuticle. This cuticle I extracted in Trizol. The sample quality of and concentration of those RNA was checked using cubic. I also checked using agarose gel. The cDNA libraries was prepared, the sequencing, and then the RNA-seq data analysis I performed in the University of Copenhagen. To do this RNA-seq analysis, we first picked the RNA-seq reads. We filtered and trimmed them. Then I mapped them with the reference genomes from Metahizum anisopi and from Metahizum riley, according to the treatments. I did the PCA plots, the diagnostic plots, and the gene count matrix before running the differential gene expression analysis. After the differential gene expression analysis, I did the annotation of the protein sequences found there. And then I did the analysis based on bibliographic reference to understand what is what on those PFAMs. So to results. Here first, if we look into methylhistone anisopli, yield of blastospores using 2 concentration of sugar, the high glucose in blue and the low glucose in yellow, we can see methylhistone anisopli responds better to the high glucose concentration. It produces more than the low one. And here 2 isolates. We have 2 isolates for those stocks, and both of them produces more and smaller blastospores. So here, besides we have the cell volume, and really we can check it, the smaller blastospores in high concentration of glucose. But in methylhistone Riley, it responds differently. It is completely opposite. So methylhistone Riley responds better in low concentration of sugar. So it produces more blastospores in low concentration of sugar, and smaller one also in low concentration of sugar. So it is the opposite. And when we go to the virulence test, first the methylhistone anisopli was not that virulent to Spodoptera frigipirida. It is not specialized in Spodoptera frigipirida, and it killed almost 25% of the population. And we didn't have any difference between conidia and blastospores. Even blastospores produced in low concentration of sugar are high. But when we look into methylhistone Riley, we had a difference. The conidia was more virulent. It is specialized in Spodoptera frigipirida caterpillars. So it has more virulence. But if we look into blastospores produced in low concentration and high concentration of sugar, the blastospores in the high concentration of sugar respond better according to our hypothesis. And here the scanning electron microscopy studies. So I have first blastospores among all those times, and we find out that 6 hours after spraying was better for the transcriptome studies because we saw the penetration occurring. So here we see blastospores with a hyphae, and here we have the blastospores with the germ tube, and we can see the blastospore turning to empty, you see? So we choose 6 hours after spraying for the transcriptome studies for blastospores, and we choose it the 24 hours for the conidia because we saw the apressoria here, you can see, and also the conidia turning to empty. So we could understand the penetration was occurring on this time. And then we did the RNA extraction of both methylhistone anisopli and methylhistone Riley, blastospores and conidia. And what we can see here is they are the opposite. The expression of them are the opposite. So while we have upregulation in conidia, we have downregulation in blastospores, and the same occurred here in methylhistone Riley. We had more differential expression genes in methylhistone anisopli than in methylhistone Riley. Usually we expect 10% of expressed genes on the transcriptome profile, and we found more genes in methylhistone anisopli than in methylhistone, compared with methylhistone Riley, and this could be explained because in generalists, usually we have the proteins family expansion, so we have more PFAMs in generalists compared to specialists, according to those authors which explored the speciation process and the gene expansion. And here we also can see the more genes expressed in methylhistone anisopli, which is a generalist compared with methylhistone Riley. We only had one gene on the middle, one PFAM, sorry, shared between Blastospores conidia from Metahizon isopli and Blastospores and conidia from Metahizon riley, which is a P450 cytochrome, which is a detoxification PFAM. So if we look into the different gene expression between Blastospores and conidia on the cardiopillars from Metahizon isopli and Metahizon riley, here this picture shows for us the different kinds of PFAMs divided into virulence factor, oxidative stress, nutrient transport, cell wall, cell function, biotic and abiotic stress. I did it using the bibliographical review. And if we look first into virulence factor, we can see more virulence factors being expressed in conidia of Metahizon isopli compared with Metahizon riley. If we look into oxidative stress, we also saw more in conidia of Metahizon isopli. The heat shock proteins are present in conidia of Metahizon isopli, and cell function more in conidia of anisopli. In conidia of generalists, we have more virulence factors related to generalists compared to the specialist one, the Metahizon riley. And this could be related because they don't have specific mechanisms to evade the roast. So it needs to produce more virulence factors compared to the specialist one. So here we look into only virulence unregulated PFAMs. And we compare Metahizon isopli, Metahizon riley, Blastospores and conidia. And what we found here, the same PFAM shared among all of them, the cytochrome P450. And we found some virulence factors shared among Metahizon isopli and Metahizon riley conidia. And we found a little bit in Metahizon isopli and Metahizon riley, Blastospores compared with conidia, but we haven't found the any gene shared among Blastospores. So here, one shared by all structures, 10 shared by Metahizon isopli and Metahizon riley conidia, 3 shared by Metahizon Blastospores and conidia, Metahizon riley, but we haven't found any Blastospores of both species sharing virulence factors. So the conclusions for this paper is the Metahizon riley is a Lepidoptera specialist fungi were more violent to Spodoptera frigidipeda compared to Metahizon isopli. However, the Metahizon isopli expressed more virulence and oxidative stress factors compared to Metahizon riley. And analyzing the difference among Blastospores and conidia, the Metahizon riley Blastospores express more virulence-related genes against Spodoptera frigidipeda than Metahizon isopli. If we look into the opposite, the conidia of both of them express more virulence-related and oxidative stress factors. So the conidia is more adapted to kill fast and produce many enzymes and many virulence factors to evade the host. And our results highlight, so the Blastospores use a different virulence mechanism. We don't know exactly how and exactly what, but we could see those differences between conidia and Blastospores and between generalists and specialists against the same host. So answering the main hypothesis I put on the first chapter. Are the Blastospores producing liquid culture media more virulent than conidia? Not exactly, because it could be more violent, the virulence could be related to which form we produce. The osmotic stress in liquid culture media improved the Blastospore production. Not exactly, because in Metahizon isopli was one thing, and for Metahizon riley we had another one. Comparing propagandist, looking into Blastospores, do Blastospores and conidia act on the same way? No, they don't act the same way. We could see difference among them. And comparing the fungal species, do Metahizon isopli and Metahizon riley act the same way? no, they don't act the same way. They also have difference during the penetration on the caterpillars. So this was the third chapter. So we are going to the last one, which we investigate the phenotypical plasticity and the transcriptome profile of Metahesium anisopli. Only blastospores here during the penetration on different insect hosts. So on the first part of the thesis, we investigated different species of Metahesium, a generalist and a specialist, and we look into different propagulists. But here we looked only in blastospores, looking into different insect hosts to see, oh, we have a generalist species, how they act. So it is not published yet, but we are working on this. And just a quick introduction, the generalist entomopathogen, they have broad host range, and they have to deal with these different insect hosts and different tissues to evade the host. So they need to tailor the expression of virulence-associated genes to specific host species. They have to adapt to different insect host tegument and immune response. So they are versatile. We can say they are versatile because they have to adapt for each insect they would evade, and they have phenotypic flexibility to evade. But they also exhibit variation on insect virulence. It is not because they can infect different hosts, they can have the same pathogenicity, they can exhibit the same virulence for all of them. For Metahesium anisopli, they are, it is one of the most studied and applied biological control agents worldwide, as I already told you. And blastospores for biological control is not useful yet. We are not using this on the field, but they can be produced in liquid culture media, and they can be used directly to infect insect hosts. We can produce it in large amounts in a short time. It's really, really, really more faster than conidia. We use a small space, only a bioreactor, and we use less manual labor comparing to the solid fermentation method. They are hydrophilic propagulists. They are easy to formulate. We don't need to use oil to formulate blastospores. They germinate faster than conidia. They germinate between 2 and 8 hours. In our last study, we found 6 hours on the germination inside the insect, but the conidia takes usually 24 hours, so it's really different between them. And these factors may be suitable for a primary propagule for using in biological control. They are less able to be present on the environment because the UV protection they don't have, but it is a thing to improve. So our hypothesis, the Metahesium anisopli can also modulate its phenotype and the gene response to adapt and cause disease to different hosts. And for this, we investigate this phenotypic plasticity and the differential gene expression of Metahesium anisopli blastospores during disinfection on different insect orders. So we did the same production, only with Metahesium anisopli here. We checked for contamination, so all flasks were checked for contamination. And we performed the virulence test with 4 different species of 4 orders. So we used crickets, the grills and similis, a caterpillar, Spodoptera frugiperda, the Apis mellifera honeybee, and bugs that Tenebro monitor. And we also did the microscopy of the propagule germination to check the penetration and to adapt the methodology before RNA expression. So we use WINS as a model for the studies of penetration. but we aim to know if blastospore is used to penetrate in wings in 6 hours as the caterpillars. So we took some criteria. The amount of blastospores on those wings must cover all the tissue homogeneous before RNA extraction, without overlapping propaguly to avoid non-germination, for example. The germ tube of those germinated blastospores must be at least the double size of the size of the blastospores to avoid non-germination also. The appressoria formation must be present in at least one treatment to guarantee the penetration was occurring at that time. So what we looked for was, here we can see the blastospores and the appressoria formated inside on the surface of the wing. So many of them. So this is the kind of imaging we were looking for, and we perform at different times and different concentration to check which one was the best for RNA extraction. We use the membranous wings of Tenebrio, Gryllus, Spodoptera, and Apis, and we only use blastospores of Metahizium isople. So the volume of the suspension and the amount of wings per sample varied according to the host. Here we can see Apis mellifera wings, and it's very tiny, but the crickets, for example, they are huge. So the process was pretty the same. Regarding here, we use the adults, so we removed the hind wings, the membranous wings, before applying. Then we apply inside these membranous wings, as this picture here. And we performed the RNA extraction, the sample quality, we performed the cDNA libraries, the sequencing, and the data analysis. The data analysis here was a little bit different because we had only one fungi, only one kind of propagule, but we had different insect orders. So after filtering and trimming the reads, we mapped only with the Metahizium isople genome. We performed the diagnostic plots and the DSEC analysis using pairwise combination. Then we also did the annotation of protein sequences. We choose only the unique differently expressed genes among all of the species. We perform enrichment analysis, and then we investigate the gene expression per host using Venn diagrams to understand it. And the last, we did the analysis based on the graphic reference. So going to results, the virulence to crickets was not that good, and we were expecting that. So despite the virulence was not that good, we could see blastospores was a little bit more virulent and differential. We had differential here in blastospores. For Spodoptera frigidata, the virulence was not that good also, but we also see difference here in blastospores, killing more than Conidia. We also did for Apis, and as the same pattern, we see blastospores being more virulent than Conidia. For Apis, it's really tricky to perform bioassays because they died a lot. So we had many deaths in the control along the time, but the blastospores were also more violent here. And for Tenebrio, we had the most virulence, and also blastospores were more violent than Conidia for Tenebrio. Summarizing those virulence tests, they was performed and they showed different mortality levels among the insect species. We saw the highest mortality in Tenebrio molitor and the lowest one in Spodoptera frigidata and crickets, for example. The same pattern could be observed for all of them, that blastospores were always significantly more virulent, and both blastospores and Conidia caused high mortality in Tenebrio monitor and Apis mellifera population was the highest virulence. And neither blastospores nor conidia were virulent to crickets and to Spodoptera. So here are the results of our pictures for germination. We studied 4 hours and 6 hours because some papers says they can germinate it with 4 hours, and yes, they can, but we could see Apressoria only in 6 hours. That's why we choose it 6 hours. So here we used 5 plus 10 to 7, the best concentration, and here we can see among those species, only in Apis mellifera we could not see at the same time the formation of Apressoria. So we could see in Spodoptera, we could see Apressoria in crickets, we could see in Tenebrio, but we could not see in Apis. And we quantified this Apressoria formation, counting the Apressoria formation in each sample. So we could see it really did not form Apressoria in Apis mellifera, but it forms some Apressoria in crickets and Spodoptera, and the most percentage of Apressoria formation was in Tenebrio, which is tricky because in Tenebrio it forms much Apressoria and kills a lot of insects, but in Apis it do not form Apressoria and kills a lot of insects. So the Apressoria could not be only related, could not be the only one related to virulence, you know. And looking into differential gene expression, so we did the DSEC analysis in pairwise comparisons. So here we have each pairwise comparisons looking into upregulated and downregulated. We performed the gene co-expression analysis using cluster analysis. So we found 7 different clusters comparing all the gene expression among those insects. And here a look into all the total genes expressed on the left. And on the right, we have only the differently expressed genes among crickets, Spodoptera, and Apis and Tenebrio. We took one of the samples of Apis mellifera because it was really different from the others. And we see here the crickets on the graphic of the left. Here the crickets, the Gryllus assimilis was differently expressed compared to the other ones. So we did the enriched epiphenes analysis, and what we can see here is the downregulated and upregulated among those functions. Those functions I separated using bibliographic references. So I divided into virulence factors, nutrient and substance transport, cell wall and membrane components, and cell function. And we can see the virulence factors here are in the low amount compared with the downregulated one. And when we look into functions, we can see most of them in cell function and virulence factor among all the insects. So those are the epiphenes found among all the insects during the germination of Metahizium anisopli blastospores. And then we look into the virulence per host. So first we look at the unique genes expressed in all of them, and then we look into each host. So for Tenebrio monitor first, which was our highest virulence, we found two different expressive genes. My pointer is not working anymore. Is it lower battery? Sorry, I will pass here because my pointer is not working. So we found only 2 related genes in Tenebrio compared with the other ones, which is the tyrosinase-like and the G-benzothiophene. And they are related to biosynthesis of secondary metabolites and virulence xenobiotic response. And then we look into each comparison, and we look into the percentage of those different expressions of the genes, and we found 1%, 1.5%. And what we found here was the MED1 and the tripeptide peptidase. And the MED1 is related to adhesion, and the second one is the subtilizine serine protease subfamily, which is related to the degradation of proteins for fungus nutrition during infection. Here, when we compare with Spodoptera, we found the hydrophobin-like protein, and we found also the strucins. The strucins are related to inhibition of Malpighian tube and suppression of insect defense response, so it's virulence related also. And comparing Tenebrio with EPIs, we found lipases and aspartic proteases related to the degradation of specific substrates and chitin proteins. And when we look into the gene expression in Spodoptera frugiperda, we found 6 genes, and among those 6 genes, we found 3 which is related to phosphodegrading proteases and degrading of chitin. So virulence related genes. So among 6 which we found here, 3 are related to virulence. And in crickets, we found 22, and most of them are hypothetical proteins, but we also find lipases, endokitinases, amidases, substrate transporters, and many hypothetical proteins. And the conclusion of this paper is the metahism really changed its phenotype, it's changed the blast scores and changed the genetic response toward different insect hosts. Expresses specific enzymes like lipases, oxidases, chitinases, sugar transporters, the strucins, and among different hosts. The blast scores of metahism were highly virulent to the nebulizer. So the most virulence we found with the nebulizer. But we didn't find any downregulation in metahism blast scores. I could not show you here, but we can find this is the downregulated genes per host also. And the phenotypic plasticity gene expression is not directly correlated with virulence in blast spores or chlamydia towards the insect we could analyze. For example, we found the absence of oppressoria inapis mellifera, but it was higher virulence. So yeah, we cannot relate the virulence to only to the oppressoria formation. And answer our questions we did on the first chapter. As a generalist, can metahism be pathogenic to different insect species? We see it can be pathogenic, but they have not the same virulence in all of them. So it can be more pathogenic for one species than to the other one. And can this fungal species adapt its phenotype and gene response to penetrate and cause disease in different pets? Yes, it could. Among the species we checked in here. So thank you. I don't know how much time I got, but I would like to thank you all, especially the University of Copenhagen, the agents that gave me money to perform this study. And yeah, thank you all.